Hi everyone, this is Chapter 15, Translation. I'm going to divide this up into three parts, and the first part is the experiments that led to the conclusion that genes encode proteins. This is a model of a ribosome. You'll notice that there are a lot of proteins, but there's a lot of RNA that is actually the structural and catalytic part of the ribosome, and we'll get into that in more detail later. But the events leading to the discovery that genes encode information to make proteins came from a gentleman by the name of Archibald Garrod. At the beginning of the 20th century, he first proposed a relationship between genes and protein production. He was studying a trait called alkaptonuria. It's a recessive trait that was due to a missing or defective enzyme. He coined the term inborn errors of metabolism. Alkaptonuria is a disease characterized by the fact that patients have black urine and a bluish-black discoloration of their cartilage due to the accumulation of abnormal levels of homogenistic acid, alkapton, and that's a consequence of improper metabolism of an amino acid. This is a pathway for the processing of an amino acid called phenylalanine. Phenylalanine is a large amino acid. You eat it when you eat all kinds of protein. And there's a pathway to break it down so that the body can recycle the components and use them. The first step after you eat phenylalanine is an enzyme called phenylalanine hydroxylase, which puts a hydroxyl group on to phenylalanine and turns it into tyrosine, which is another amino acid. From there, tyrosine is modified by tyrosine aminotransferase into para-hydroxyphenylpyruvic acid, and then another enzyme changes it to homogenistic acid, and then another enzyme changes homogenistic acid into malonyl acetic acid. Okay, if the enzyme that changes homogenistic into malonyl acetic acid is defective, if this enzyme is defective, you build up too much of the homogenistic acid and you have the condition called alkaptonuria. There are other conditions that have been identified with mutations in other enzymes in this pathway. If you mutate this enzyme, you'll end up with tyrosinosis, too much tyrosine. And if you mutate this enzyme, phenylalanine hydroxylase, you end up with a very serious condition called phenylketonuria. Phenylketonuria leads to severe cognitive and motor functions, abnormalities, so much so that every newborn in the United States is tested for phenylketonuria by a very simple blood test. Anyway, Archibald came up with the idea that if you have a mutation, you can identify a single enzyme in a pathway. Okay, so Beadle and Tatum came along. Beadle and Tatum, obviously very famous scientists. Here they are dressed up for their Nobel Prize receipt at the dinner. They worked on an organism called Neurospora crassa. Here it is lit up with a green fluorescent protein. When Neurospora crassa undergoes its life cycle, undergoes its undergoes its cell division, it produces a sporopod that has 8 different spores in it, and they divide right next to each other so that the first 4 spores come from one cell and the second spores come from another cell, and you can actually see whether or not they have the same mutations in all of the cells. In this case, in this pod, these 4 cells are identical but different from these 4 cells because these 4 cells are expressing green fluorescent protein. This is what it looks like when it's growing in a Petri dish. It's a quite common bread mold. Okay, so in the early 1940s, they asked this question: is it just one gene and one enzyme, or is there one gene that produces many enzymes? Their genetic model, like I said, was Neurospora crassa, common bread mold, and their studies involved analysis of simple nutritional requirements. If you can remember, prototrophs are organisms that can grow on very simple nutrients such as a carbon source and simple salts, whereas auxotrophs require some other more complicated molecules to be included in their nutritional supplements. So they were interested in following similar kinds of pathways that Garrod had analyzed. In the process of amino acid biosynthesis, in order to end up with a final functional amino acid such as methionine, you have to start out way back here with a precursor product that gets acted on by an enzyme that turns it into another precursor, that should be two, that gets acted on by another enzyme that gets turned into a third precursor, that gets acted on by another enzyme that gets turned into a fourth precursor, that gets acted on by another enzyme until you have the final product. What happens if there is a mutation in one of these enzymes in the pathway? Well, if you knock out enzyme 2, that means you'll build up precursor number 2, but you won't make the final product. Okay? You'll also have an abundance of precursor number 1. So Beadle and Tatum said, okay, we're going to mutate our Neurospora and see if we can identify all the different enzymes in a pathway and order them. So they took some Neurospora and they hit it with X-rays and they isolated some, and then they let them develop into a sporocap, and then they isolated the different spores to find out what the mutations were in each of them. How did they determine what the mutations were? Well, they made some minimal media plates supplemented with single amino acids. So the minimal media is essentially salt and sugar. And then one by one they added back amino acids or their precursors. They divided plates into 5 segments, and on each plate they streaked out a little bit of a different mutant strain. So here's what the experiment looked like. Here's a minimal media plate and divided into 5 different segments. In this segment, in the last segment, they put wild-type Neurospora. And on minimal media, wild-type had no problem growing. However, none of these mutants would grow. They needed to be supplemented with something else. And that's what all these plates are. These plates have been supplemented with different precursors in the amino acid biosynthetic pathway that's going to produce methionine. So they added acetylhomoserine, cystathionine, homocysteine, and finally methionine itself to each of these plates differently, not all together, but each one of these plates had a different supplement. They made note of which strains grew when supplemented with a particular amino acid or precursor, and then they assembled the biosynthetic pathway. So they came up with this very simple pathway for methionine biosynthesis. If homoserine was the initial precursor to the entire pathway, when they added acetyl homoserine, strain number 1 grew, but not strains number 2, 3, and 4. So they concluded that strain number 1 had a defect in enzyme number 1. If you gave the fungus some of the product, they didn't need enzyme number 1, and enzyme number 2, 3, and 4 were just fine. So as long as you had the acetylhomoserine, you could make cystathionine, homocysteine, and then methionine. Okay, what about adding cystathionine? Well, if you add cystathionine, then strain number 2 was also okay, whereas when you only added the acetylhomocysteine, it couldn't grow. Also, the strain number 1 grew because its defect was in enzyme number 1. Okay, finally, of course, if you add methionine, all of the strains grew because that's the final product and that's what all of these other enzymes are supposed to be making. Okay, that's it for the first part. The next part is going to be reading the genetic code.